let's talk. We start talking about metabolic strategies in the microbes. So the way how microbial cells collect energy and nutrients from the environment. Okay, and I told you before that scientists love to put things in different boxes. So different metabolic strategies provide us with a great way to classify microbes. First, before we move on, we have to define what metabolism is. Anyone? What is metabolism? Okay, so breaking down, is that it? Breaking down, absolutely. Breaking down molecules, large molecules to release energy. What else do cells have to do? What? Well, it's one of the ways to break down glucose. What else? Excellent. Take small ones and put them back together, okay? To grow. So essentially, if you think about what Chris just said, it's a bunch of chemical reactions. So metabolism is the chemistry of the cell, right? And it can be, so reactions when cells break down large molecules to the smaller one, called catabolic, the reaction of degradation. When smaller molecules are collected together and form larger ones, are anabolic reactions. There are billions of reactions going on at any given moment in a cell. But it turns out with the development of current technology, we can take a cell and pretty much determine almost all chemicals that are in the cell at any given time, and it is called, this approach, overall approach is called Metabolomic Studies. So the science, modern science, moves more and more to accumulating vast amounts of data. You see the difference? Instead of asking a very specific question and trying to answer it, I don't know, finding a specific enzyme or a specific product of reaction, scientists now can take a cell, lyse it, run it through, say, the chromatography machine, and then analyze the data and figure out, okay, cell in these conditions produces these lipids, this carbohydrates, this, I don't know, that, okay? And then they look at this data and say, so what? So what does that mean? What this metabolome means? Or they can take cell and put it in one set of conditions and discover the metabolome, put it in another set of conditions, discover another metabolome, and then compare them and see what changes in very many aspects instead of measuring just one. So you probably have heard the statement, like the name big data. That's what scientists work with now. They work with big, huge amounts of data, and of course it's not humans that compare them hand by hand, it's computers that do that. Now, going back to classification based on metabolic strategies, all microorganisms, and not only microorganisms, need two things. They need energy, and they need organic carbon. Okay? Because why do they need organic carbon? Why organisms need organic carbon? Yeah, they're made of it. They needed to build themselves, to grow. Essentially, organisms need carbon as a source of nutrients. Where do we get our carbon from? Food. Trust me, all food that we consume, starting from Papa John's pizza and ending Whole Foods apples, I don't know, $15 a pound, it's all organic. It's all organic. It all contains organic carbon. From the chemical standpoint, it's all organic. Plastic that this computer is made from is organic. Okay? So, chemically, for me, there's only one organic. It's chemically organic. Now, based on the carbon source, we can separate organisms, all organisms, whether they take carbon from inorganic or organic sources. So we figured out that organic sources of carbon can be food for humans. What about other organisms? Where else can you take organic carbon? It's not organic. That's carbon dioxide will be a first choice of inorganic carbon. What about organic one? Okay, well, sugars, but I mean in the environment, look, not literally, but figuratively, look outside the window. Where can you take? Huh? Okay, living organisms. Can we take carbon from living organisms? The other organisms that take carbon from living organisms, how do we call them? Parasites. Yeah, those are parasites, right? Where else? What about dead organisms? Sure, right? Decomposition is perfectly fine. So you can take organic carbon from living creatures, from dead creatures. Fungi, to be honest, they take organic carbon from pretty much anything, starting from oil and ending with plastic. There's a joke that was only partially a joke. If there is a source of organic carbon, there's going to be fungus that will use it. They are absolutely unique in such adaptations. So again, organic sources are vast and multiple. Inorganic are limited. It's either carbon dioxide or in some cases it can be bicarbonate. Okay, but it's all inorganic. Mostly it's carbon dioxide. Now, along with the sources of carbon, organisms need to get energy, right? There are two major sources of energy. One is sunlight. Very good. And another is chemical source of energy. Now, chemicals that can be used as the energy source can be, again, organic, or in some cases, they can be inorganic. Now, this may sound again like a little bit of a philosophical question, but let's start with sunlight. So sunlight, which organisms use sunlight as the source of energy? Plants. Okay, let's take plants. So they use energy. What they do when they use energy? They grow, right? They grow, okay. Now, when they grow tall enough, what happens to the plants? Very frequently. They reproduce, but do they like die peacefully or? Yeah, well, I mean, but look outside. Unless we cut the grass, it's going to grow tall. But there's another way to limit it. You can put cows on the pasture, okay, and they're going to eat it. When cows eat this grass, what's going to happen to cows? Huh? They're going to grow, right? They're going to become bigger. And what happens to the cows normally in the end? We eat them. And when we eat them, we grow. If you think about this, this entire chain is about energy transformation. plants convert energy of light into the chemical energy of the bonds in a starch, right? And cellulose. Cows eat plants and convert chemical energy in the starch and chemical energy of cellulose into ATP, which then they use to produce proteins, meat. We eat meat and we break down meat, and chemical energy of peptide bonds in the protein is used to produce ATP in humans, which we use to grow. It's all about energy conversions. Does that make sense? So if you think about it, the entire life is transforming energy, either from one chemical form to another chemical form, or from the form of light into the chemical energy. Or we can also convert it into the mechanical. When we run, okay, we convert chemical energy of ATP into the mechanical energy of movement. Does that make sense? Now, a little bit about chemical bonds. So if you have like two molecules or two atoms connected by the bond, if you break it, energy will be released. Does that make sense? Now, if you form it, energy will require to form the bond back. Does that make sense? Very basic, okay? Does that make sense? So we figured out two things. First, nutrient sources, right? Can be organic or inorganic carbon. And second, energy sources. Basically, it's either chemical energy or physical from light. So organisms that use light as the source of energy are called phototrophs. Okay? Organisms that can use inorganic sources of carbon are called autotrophs. So plants that use light as the energy source and inorganic carbon from CO2 called photoautotrophs. There are microorganisms that use light as the energy source but utilize organic sources of carbon. They are photoheterotrophs, certain types of bacteria. Okay? Now, we are Chemo Heterotrophs. Chemo points at the source of energy. What is the major source of energy for humans? Which nutrient? Glucose, yes. We can use others, but it's mostly glucose. And source of carbon, everything that we eat. Okay, but it's all organic. We don't inhale CO2 and convert it into the organic matter. We don't drink baking soda and then this carbon magically incorporates in a human body. Okay? There are, though, Chemo Autotrophs, which I find absolutely fascinating. So they use chemical reactions as the source of energy, and they use inorganic sources of carbon, like CO2 or the carbonate. Now, they are also called lithotrophs because the chemical source that they use is not based on the organic matter. For instance, some of them, let's say sulfur oxidizing bacteria, will essentially perform this type of the reaction. They will oxidize, well, strictly speaking, sulfide ion into the sulfate ion. Okay? So the electrons will go from sulfur to oxygen. Okay? Some of them can oxidize ammonia and convert it into the nitrates. Right? So it's all about energy. Does that make sense? Yes.
Are those like the ones that are found at the bottom of the sea next to the vents?
Yes, definitely these guys. Sulfur oxidizing, yeah. They one statement is not 100% correct about bacteria. A lot of them are archaea. But I wouldn't focus on that much. I mean, it's prokaryotes, okay? Another thing that I want to point out here is which elementary particles form chemical bonds? No. Elementary particles. Like proton, neutron, electron, electrons, right? Electrons form chemical bonds. So if you think about chemical conversions in the organisms, it's all about electrons transferring from one molecule to another. So it's an electron flow. Electrons are, I don't know how to say it, medium. Electrons carry energy. Does that make sense? So when electrons are transferred from one molecule, that gray circle is electron. This electron is transferred from that blue sphere to that purple cube. Okay, you see the purple cube now has an electron, while the sphere is stripped of electrons. How do we call these reactions? Redox, yes. Reduction and oxidation. You have to appreciate the fact that these two cannot happen without each other, right? Because when molecule gets stripped of electrons, what happens to it? How do we call it? It gets oxidized, right? When molecule gains electrons, it gets reduced. Now, the molecule that gets reduced is oxidizing agent. The molecule that gets oxidized is the reducing agent, right? Or we can call this an oxidant, and we can call this a reductant, right? Just that's simply English language, okay? Whatever the molecules do, right? So it's electron move from one molecule to another. Can you give me the simplest example of oxidation and reduction that you may observe? Huh? Say again? Rust. Excellent. So iron gets oxidized and becomes iron oxide. That make sense? So that's a perfect example of oxidation. Oxygen takes electrons away from the iron, right? So iron is oxidized, oxygen is reduced. Now electrons that go from one molecule to another, we can say that they carry energy, and that will be true. And we can, I usually illustrate it this way. So you have electrons in one molecule and electrons in another molecule. And as they go from one molecule to another, they release energy. Another example of oxidation and reduction is when you burn something, like burn paper or burn coal. Okay? It releases a lot of energy. Does that make sense? So again, the chemistry of life, the energy transformations in the cell are all about electrons flowing from higher energy level to lower energy level. And as they flow from higher energy level towards lower energy level, they release energy. Of course, not. Now, this energy, what do we use it for? What cells use it for? Microbes, what they use it for? Huh? See again? Whatever the cell needs. Energy consuming reactions like synthesis. Okay, like like translation, transcription, replication, I don't know, synthesis of biochemically important molecules, storage of nutrients when they pack them in the form of the polymer. Does that make sense? Energy is not, well, it is wasted to an extent, but cell attempts not to waste it. Okay? Now, electrons are extremely chemically reactive. Have you heard about the radiation? I suppose you have. There are three types: alpha, beta, and gamma. Beta radiation is electrons, and it's pretty badass. Okay, it can ruin the cell. Okay, so free electrons is impossibly reactive. Okay, now you don't and say you have, especially in like in places like a eukaryotic cell, you have glycolysis going on in the cytoplasm, and electron transport chain is in mitochondria. You definitely don't want electrons to travel by themselves from cytoplasm to mitochondria. You want them to be bound to something, like they should take a taxi. Okay, they should be carried, and molecules that carry electrons are quite surprisingly called electron carriers. One that you definitely know is NAD. Okay, it's nicotinamide adenine dinucleotide. It's a tongue breaker. I ask you to understand and know the acronym rather than the entire name. So NAD can receive 2 electrons. What is the charge of electron? Negative. So to balance the negative charge, NAD also binds 2 hydrogen ions that are positively charged, right? And what you end up in the end, when NAD accepts the electrons, is it oxidized or reduced? It is reduced. So reduced form of NAD is NADH, and there's a proton, the hydrogen ion that travels near to it. Does that make sense? Another molecule that you also might have heard about is FADH. For FADH, it can accept electron and hydrogen ion, and its reduced form is going to be FADH2. Does that make sense so far? So NADH and FADH2 will carry electrons between different parts in the cell. Right? Now, all this talk about energy conversions, what is the ultimate goal of the energy flow in the cell? What is the final product of energy conversions? What is the molecule that provides energy for everything in the cell? ATP. Did you ever wonder why? Did you ever think why? I mean, there's no why question in biology, but nevertheless, why ATP? Why we can't just simply use glucose as a source of energy, or lipids, or both? Any ideas? Well, think about this. You have a molecule of glucose, right? And you put it into the oxidative phosphorylation. Some of the energy will inevitably be lost as the heat. And then ATP hydrolysis is not 100% effective. So if you add more and more steps, each step doesn't have 100% efficiency. So in terms of efficiency, it's easy, but you have to invent like the whole new system of enzymes for ATP synthesis, for ATP breakdown. What if we get rid of all the money? So say one person knows how to fix cars, another one knows how to bake bread, okay? And I need strut replacement. So I go to that dude and tell him, look, I need strut replacement. He replaces the struts. And I give him 50 bread loaves. I mean, it's probably a good deal for me. Just for 50 bread loaves, I get struts replaced. But honestly, what this guy's going to do with 50 loaves of bread, I have no idea. He needs to sell them or exchange for something else. So that natural exchange becomes a pain in the butt, right? So we invent money. Money is the currency. Money value everything, right? Now think about this. Imagine that you decide, okay, I'm going to get rid, well, hypothetically, I'm going to get rid of ATP. So now you have translation, which requires a lot of energy, and you have a certain enzymatic system that extracts energy from glucose. You run out of glucose. Now you have to have a dedicated system for lipids, not only to extract energy, but to channel it directly to whatever is needed during translation. So those systems have to interact with each other. Separate system has to have, you got to have it for replication or transcription or some other form of synthesis in the cell. So things become extremely complicated. Instead, you have a dedicated system that produces ATP. And you have a dedicated set of enzymes, ATPases, GTPases, that participate in all energy-consuming processes by breaking down ATP. Does that make sense? It's a currency. It's really a currency in the cell. So you don't have to, well, not you, but there is no need in developing so many metabolic systems for every task. Does that make sense? Now ATP stores energy in the form of a chemical bond. Gamma and, to a less extent, beta phosphate bonds are energy rich. Gamma phosphate bond is the most energy rich. You break down ATP, you get an organic phosphate and ADP, and there is energy released. But in order to make ATP back, you got to use energy. Does that make sense? So you got to use energy to produce ATP, which then can release energy that's necessary for intracellular processes. Reactions that release energy, called exergonic. That's catabolic reaction, mostly. So catabolism is degradation. Can you think of a catabolic reaction right off the bat? Breaking down something. Protein catabolism, breaking down proteins to amino acids. That's catabolic reaction, and it will release energy. Because that's what the breakdown is for, right? ATP breakdown, ATP hydrolysis is catabolic reaction, which releases energy. Right? Anabolism is mostly endergonic, which means it requires energy. Now, every time you see a new word, think about its structure. And if you can't get the structure, ask me if I don't explain it. So, in case of exergonic and endergonic, erg, E-R-G, usually refers to energy. Okay? That's energy. Now, X, exit, out, and in. Does that make sense? Exergonic, energy out, and ergonic, energy in. Does that make sense? Again, those reactions are coupled. Energy doesn't just go out, okay? For instance, when ATP is hydrolyzed and energy is released, it's catabolic. This is catabolic, no, wait a minute. Catabolic and exergonic reaction right here. Hydrolysis of ATP. This energy can be used for synthesis of, I don't know, proteins or polysaccharides. But for ATP synthesis, which is endergonic and anabolic reaction, energy will be taken from, say, metabolism of glucose, catabolism of glucose, oxidation of glucose. Does that make sense? Now, when we get to the end of this chapter, we will talk about photosynthesis, and you will see that during photosynthesis, light, even light, it produces high energy electrons, which energy is then stored for short term in the form of ATP, that is, and those electrons are on NADPH, and then those electrons are used to form carbohydrates. So energy is stored in the form of chemical bond in carbohydrates. And then when we eat those carbohydrates, we release that chemical energy and convert it into the ATP. Energy does not ever go in vain. I mean, it does. We warm-blooded animals, that's where it goes, okay? But generally speaking, energy is used for something. Energy from one reaction is used for another. Does that make sense? Okay? That link, catabolic, anabolic, exergonic, endergonic. Right? I'm going to write you the reaction. What's going to be the product of that chemical reaction? Water. So what is this? How do we call this? What is that? Huh? Hydrogen. Yeah, hydrogen. What about this one? Oxygen. Hydrogen and oxygen. What physical form do they have? They're gas, both of them. So you can mix them, right? I mean, you can get pure hydrogen, you can get pure oxygen, you can mix them in like a tank, say glass tank. So you mix them. What you're gonna see? You wish. Nothing. Huh? You will see condensation if you live long enough. We're talking 1,000 years. So this reaction doesn't go by itself. Or, better yet, I used to have, okay, matches. Potentially, can they burn? What burns them? Why they burn? With what? Oxygen in the air. Do I have match? Do I have oxygen? Nothing happens. Why? I need what? I need to apply heat. So I apply heat. Do I need to apply any more heat now? No, it goes by itself, right? But I needed it in the beginning. Why? To create reaction. Yeah. So I need to activate the reaction. It's called the activation energy of the reaction. It's kind of a bump that you have to go over to start the reaction. Does that make sense? Now, without, yes.
Pressure would work as well as heat, wouldn't it?
Depends on the reaction. Like if you talk about diesel engine, then yes. In diesel engine, you mix oxygen, well, air, and diesel fuel at the very, very high pressure in a cylinder. And that pressure increases temperature right away. Does that make sense? And it starts to burn. Okay. For hydrogen and oxygen, probably so. It probably would work. But look, if you mix hydrogen and oxygen and you put the match in it, you're going to die. Because this thing is going to explode and it will kill you. Really effectively explode. Okay, if you ever heard about the Hindenburg, one of the Zeppelins, it was filled with hydrogen and went boom. Okay, so in order to start the reaction, you have to go over the bump. You have to go over that activation energy point. Okay, and as I just told you, these reactions, chemical reactions, without proper activation, may take 100 and 1,000 years to happen. Even if they take minutes, like rust, okay? It's not 1,000 years, but it's not immediately, okay? Cells can't afford it. They have things to go in milliseconds. Evolutionary, we now got enzymes, okay? Basically, enzymes reduce activation energy. Does that make sense? Now, when they reduce activation energy, do they alter the ratio of the products? They do not. That is a fundamental feature of the enzyme. Enzyme changes the rate, but not the ratio. Bless you. Does that make sense? So if we would have hypothetical enzyme that would speed up this reaction, okay, we would still, if we mix 2 moles of hydrogen and 1 mole of oxygen, we would still get 2 moles of water, not 3, not 4, okay, still 2. Ratio is going to be the same, but the rate of the reaction will be much higher. Does that make sense? Most of the proteins are, most of the enzymes are proteins. We mentioned some that are not RNA enzymes, so-called ribozymes that are responsible for splicing and also can be found in ribosomes. Okay, but that's rare. Most of the enzymes, at least the enzymes that we're going to be talking about, are proteins. Okay. So enzyme function is to facilitate the reaction. Okay. They do not directly participate in it. So if you start with 10 molecules of enzyme, how many molecules of enzyme are you going to have in the end of the reaction when everything is converted? 10, yeah, same amount. They're not wasted, okay? The basic reaction is substrate is converted into product, and this reaction is catalyzed by enzymes. So please know what the substrate, what the product, okay? So substrate, which you can see here, this green figure, binds to the molecule of enzyme, and it binds to the place in the enzyme that is called active site. Now, active site, due to its unique shape and arrangement of amino acids, active site facilitates whatever's going on, okay? Whatever chemical reaction it catalyzes. And now you have products. Enzymes are extremely specific. There are 2 models that I would say mutually complementary, okay? Lock and key model, well, that's kind of obvious, you know. Every key is specific to its own lock. And second is induced fit model, which means that when substrate interacts with an enzyme, it changes the shape of the active site. Okay, so you see they're not equal. Okay, it changes the shape of the active site and then binds. It's still sort of lock and key. How specific enzymes are? They're enzymes sucrase and lactase. Sucrase breaks down sucrose. Lactase breaks down lactose. Sucrose and lactose are fairly similar in terms of the chemical structure, but enzymes can recognize and distinguish them. Does that make sense? They are extremely specific. Now, enzymes have optimal conditions to work. What are the optimal conditions for, say, DNA polymerase in your cells, pH-wise? 7-ish, 7.4. Temperature-wise, 37 body temp, right? What about enzymes pepsin in the stomach? pH 2, 4, 3, acidic, right? Does that make sense? Does that mean that pepsin is not going to work at neutral pH? Yeah, not as well. Oh, maybe not. We don't. But when we say optimum conditions, we mean there are. So if we would plot hypothetically the enzyme activity versus, say, pH or temperature, okay, it would look like this. So there's optimal temperature or optimal pH for the enzyme to work. It will work at others. Okay, different enzymes will have different shape of that curve. For some, it may be like this. So it's going to work at pretty much any pH or any temperature with slightly different efficiency. Does that make sense? Question? Okay. Few words about that graph over here. So, this green stuff is energy. So green line here represents the energy of the reactants, okay, substrates, okay, and green line over here, which is dashed, is the energy of products. Now, when we see this, that reactants have a lower energy than products, what does that mean? Huh? Reaction is endergonic. Very good. You see, we not only have to spend energy to go over that bump, we have to add energy to the chemical system to make products. This difference is called, if you're curious, enthalpy, which is a fancy word that says you how much energy you have to spend in the reaction. If, let's say, we will run the reverse reaction from here to here, this will be amount of energy that will be released. Does that make sense? Now, huh? Yes. Yes, in opposite process. Yes. So you, exactly, in endergonic reaction, you kind of conserve this energy, you store this energy in the form of chemical bonds. And then you break those chemical bonds and energy gets released. Does that make sense? Now, this just shows you that was activation energy in the beginning, before the enzyme was added. This one. And this after the enzyme. That makes sense? It just reduces that bump. I'm very proud of my analogy for the enzyme work. You've read Cinderella. You know, in order for her to go, I don't know, I read Russian version, which is not very different, you know, conceptually from original one. Maybe these ridiculous tasks that stepmother, you know, forced Cinderella to do slightly different. So one task was something like to sort out beans or seeds or some other crap. Okay, so pretty much stepmother mixed say white rice and brown rice. It's kind of some sort of an Indian Cinderella. So white rice and brown rice, and told her to separate those. Can you do it? Yes. How long would it take? A lot of time, right? Well, she had little helpers like mice or some other small animals, and they separated it pretty quickly, right? And then they went away. Mice were enzymes. They did the same process that Cinderella could do by herself, but much faster. And as far as I know, Not a single mouse died as the result of this process. Okay, so they all survived. 5 mice in the beginning, 5 mice in the end. Does that make sense? So think about enzymes as helpers in direction. Okay, ratio is not changed, but the rate is. Got it? Now, enzymes can be inside of the cell, and in this case they are called endoenzymes. Those would be pretty much the enzymes that are necessary for intracellular processes. Like RNA polymerase is endoenzyme, right? Enzymes that participate in glycolysis. All enzymes that do intracellular work are endoenzymes. And microbial cells can also produce so-called exoenzymes. Exoenzymes are released in the environment. What for? They're not going to be able to grab. Yes. So you two got to the right answer. So say, have you seen mold growing on the apples? I don't think that cells of this fungus actually take pieces of apple in. I strongly believe that they destroy apple first into the simplest organic compounds like glucose and stuff, okay? So fungus releases exoenzymes, and look, apple, when it's infested with fungus, becomes like soft and squishy. So those exoenzymes, they break down the sugars to the starch into the monosaccharides, which then can diffuse into the fungal cell. Does that make sense? That's all very romantic. Fungus, apples. Exoenzymes can serve as the virulence factors. We will talk about Staphylococcus aureus. You've heard about that microbe, I bet. And I don't know if you've heard about the scalded skin syndrome, triple S. So scalded skin syndrome is not very frequent, but I mean, it's not like every second child gets it, but it can happen in kids. It's a common infection of newborns when, quite literally, epidermal layer of the skin becomes slowed off, starts to fall out. It is a bacterial infection caused by Staph aureus, fairly easily treated with antibiotics. But the reason is quite exciting. If you think about skin structure, you have 2 layers. Okay? Well, you can count more than 2, but you have epidermis. Sorry, you have dermis. Okay? Epidermis is epithelial tissue. Dermis is connective tissue. Epithelial and connective tissues are always separated by the basement membrane. Staph aureus produces protease, the enzyme that breaks down proteins. And essentially, this enzyme breaks down basement membrane. So epithelium simply just falls off. Yeah? Very ouch. So that exoenzyme acts as the virulence factor. Does that make sense? There are multiple other examples, but that's just one of them. Okay? Now enzymes, some of the enzymes are produced constantly. Okay? This is called constitutive enzymes. Does that make sense? Constitutive means constant levels. And some, I would say most, are regulated. Now if you think about this, remember operons we talked about? Essentially, enzymes that are encoded by the structural genes of the operon are regulated ones. Think about inducible operon, lactose operon. When lactose is present, what happens? Binds to the repressor, repressor is gone, and what happens next? Huh? Say again? No, first transcription, then translation. Well, gene expression. Enzymes are produced. So, in this case, lactose, the substrate of those enzymes, stimulates their production. Does that make sense? Most of the enzymes are regulated. Okay, so you can see here, that's the illustration of the constitutive, when amount of substrate doesn't really do much. And this is the regulated enzyme. You see, you start off with just a little bit, but if the amount of substrate increases, the amount of enzyme increases. That makes sense. Regulation, that's going to be the last thing that we're going to talk before the break. Now, what should we start with? Let's start with the concept of apoenzyme, holoenzyme, and cofactor. So this is, let's start with this. This is apoenzyme. This is cofactor. It's Batman and Robin. Okay? So this is Batman. And this is Robin. So cofactor is the sidekick. The only difference is that apoenzyme cannot actually function without cofactor. Okay? So Batman in this case is so pathetic, he cannot do anything in terms of the crime fighting without Robin. That makes sense? So when you combine apoenzyme and cofactor, which can be a lot of things, you have a holoenzyme. Cofactors for the enzymes can be ions. Remember DNA polymerase that we talked about, that copies the DNA strand during replication? Cofactor, absolutely necessary cofactor for DNA polymerase. Just FYI, is magnesium ion. It cannot work without it. Right? Some enzymes, I don't remember right off the bat, but if you take vitamins, any vitamin, pretty much any vitamin, is a cofactor for a certain enzyme. Does that make sense? So without the vitamin, the activity of that enzyme will be inhibited, will be diminished, and that causes pathology. Does that make sense? So please understand the difference between apoenzyme and holoenzyme. That apoenzyme plus cofactor becomes holoenzyme. And that apoenzyme without the cofactor is pretty much dysfunctional. Okay? So apoenzyme plus cofactor is the holoenzyme. RNA polymerase. When we talked about transcription, I told you there are 5 subunits. So you have 2 alpha subunits, beta and beta prime subunit, and these 4 form apoenzyme. Alpha, alpha, beta, beta prime, it's the apoenzyme. But when you add sigma subunit, which is cofactor, they form holoenzyme. And interestingly enough, question? No, go ahead.
I was watching a video on transcription that says that you have to have the sigma factor in order for the
Binding for initiation, yeah.
Otherwise it
With
won't find the
it will not find the promoter, it will not bind to the promoter. The funny thing, once it starts elongating, it ditches the sigma. Okay? We actually, I worked in college, I worked a little bit on E. coli RNA polymerase. I studied it, and we stored apoenzyme and sigma separately. So in order to do things with it, so we could study, do we have to have sigma? Do we not have to have sigma? So you have literally 2 tubes, you mix them in a certain proportion, and that will form holoenzyme. Now, that's the explanation of cofactors and holoenzymes. Now, enzymes can be inhibited or activated. Let's start with an inhibition. Now, here's an inhibition. It can be competitive or non-competitive. So competitive inhibition is when inhibitor competes with the substrate for the active site. Okay? So let's say Jeff and I, we want to sit on the same chair. Okay, that chair. That would be competitive inhibition. Does that make sense? Whoever gets there first. Non-competitive inhibition is, say, I am so repulsive that even though I am here, Jeff physically cannot stand my presence this close and cannot sit on this chair. That's non-competitive inhibition. In case of non-competitive inhibition, the inhibitor binds to so-called allosteric site, which changes the shape of the active site and prevents the substrate from binding. Does that make sense? Now, you can imagine that think about competitive inhibition. If I add a lot of inhibitor, it will block all binding sites, right? But if the amount of substrate will overwhelmingly increase and just will be like 100 times more than inhibitor, then inhibitor will have very little effect because it's about competition, right? In case of non-competitive inhibition, it's not that direct. You may have very little non-competitive inhibitor, and it does not depend on the amount of substrate that you have, the inhibition rates. Some antibiotics work as competitive inhibitors. Have you heard about sulfa drugs, sulfanilamide? Sulfanilamides are competitive inhibitors of one of the enzymes in the synthesis of the vitamin called folic acid. Okay. Now, another name for non-competitive inhibition is allosteric inhibition, because it's a different site. It's not an active site. But it does not have to be inhibition. In some cases, it can be allosteric activation. When a chemical and a molecule, can be a small molecule, it can be an ion or something else, binds to the allosteric site and activates the enzyme. Does that make sense? No?
How is it inhibitory?
Say again?
How is it inhibitory if it's promoting the...
No. I'm saying it's so allosteric can be either inhibition or activation. Okay? Sorry, it's probably I just wrote over it. Does that make sense? Moreover, in some cases, the product of an enzymatic reaction can be an allosteric inhibitor. Look at this system step by step. So this is the substrate right here, which is converted to the some sort of an intermediate in direction 1 by the enzyme number 1. Then enzyme number 2 converts this intermediate A into the intermediate B. And intermediate B is converted by the enzyme number 3 into the end product. Before we move on, that's a very important illustration. Quite rarely, the reaction in a cell exists by itself. In the vast majority of cases, enzymatic reactions are the parts of the pathways. Do you see what I'm saying? That's the like a manufacturing, okay? When you go to the large plant, there's one step of the process that is linked to another step in the manufacturing process, to the third, okay? So these pathways can have absolutely different forms. They can be linear, like glycolysis. They can be cyclical, like Krebs cycle. They can branch. We can say probably fermentation. Some of the examples of heterolactic fermentation will be branch. Okay, does that make sense? Now, if you accumulate too much of a product, it may bind directly to the enzyme, first enzyme in the row, and inhibit it. Inhibit entire pathway. Does that make sense? Because when you inhibit that first enzyme, which is essentially this one, okay, the accumulation of the product will decrease. When there's less product, there's less inhibition. The enzyme, bless you, starts to be active again. So it's again negative feedback, teeter-totter. Okay, more product, less enzyme activity. Less product, more enzyme activity. Does that make sense? Okay? We're going to take a break.